Assalamu alaikum. Today I am going to discuss about enzymes part 2. Mode of action of enzymes. Enzymes speed up the rate of reaction by lowering the activation energy of reaction. Activation energy. Activation energy is defined as minimum energy to start a chemical reaction. Free energy, the energy in a physical system that can be converted to do work. Transition state. It is defined as the state corresponding to the highest potential energy along the reaction coordinate. As you can see in this graph, how enzyme reduce the rate of activation energy and boost up the rate of chemical reaction. How does enzyme reduce an activation energy? When the enzyme and substrate are in close proximity in the correct orientation, they form an enzyme substrate complex. The unstable transition state configuration state is attained. Enzyme active site provide a microenvironment that favors the reaction. Since the transition state is highly unstable, then without the enzyme, the activation energy is reduced. The catalytic group facilitate the reaction, making or breaking bond in substrate. The product is formed and released from the active site. You can see in this picture how substrate attach to enzyme on active site and form enzyme substrate complex to perform function. Nature of active site of enzymes. The active site is the region that binds the substrate. It is a region within an enzyme that fits the shape of substrate molecules. It contains the residue that directly participate in making and breaking of bonds. The interaction of enzyme and substrate at the active site promotes the formation of the transition state. The active site is the region that lowers the free energy of reaction, resulting in enhancement of the rate of reaction. Common features of active site of enzymes. Enzymes differ widely in structure, specificity, and mode of catalysis. Yet, active site have some common features. The active site is the three-dimensional cliff formed by the group that come from different parts of the amino acid sequence. They exclude water. The active site takes up the relatively small part of the total volume of an enzyme. Enzymes are so big because of scaffolding, regulatory sites, interaction sites for other proteins and channels. Substrates are bound to enzyme that multiple weak attractions such as electrostatic attractions, hydrogen bonds, van der Waals, and hydrophobic interactions. The specificity of binding depends arrangement of atom at the active site. Allosteric enzymes and allosteric effect. Allosteric enzymes are enzymes that change their conformational assemble upon binding of an effector, which results in an apparent change in binding affinity at a different ligand binding site. This is known as allosteric effect. The binding of ligand to one site on a protein molecule in such a way that properties of another site on the same protein are affected. Some enzymes are allosteric proteins, and their activity is regulated through the binding of an effector to an allosteric site. You can see in this picture, enzyme have two sites, active site and allosteric site. Substrate can attach on active site while allosteric effector can attach on allosteric site. Allosteric regulation of enzymes. Cells tightly regulate their enzymatic reactions, allowing necessary reactions to proceed while inhibiting unnecessary or damaging ones. One means of regulating enzyme action is allosteric regulation, in which a molecule binds to enzyme at site other than the active site and change the enzyme activity. Such molecule is an allosteric regulator. Some allosteric regulators activate enzyme while others inhibit them. Feedback mechanism, a process in which level of one substrate influences the level of another substance. Many enzyme catalyzed reactions are carried out through a biochemical pathway. In these pathways, the product of reaction becomes the substrate for the next reaction. At the end of the pathway, a desired product is synthesized. Negative feedback mechanism. Each metabolic reaction is regulated by several enzymes. A substrate that is affected by enzyme 1 forming product A, which then act as substrate for enzymes 2 forming product B, which then becomes substrate for the attack for enzyme 3 forming final product. As the final product becomes elevated, the system imposes a halting effect and enzyme 1, slowing down the production of intermediates A and B, reducing the formation of final product. When level of product fall, the effect of negative feedback diminishes and enzyme 1 is reactivated. Positive feedback mechanism. In contrast to negative feedback, positive feedback occurs when an output is used as a signal to increase further responses of the output. In other words, if process A results in consequence B, B reinforces process A, resulting in cascade where more of B occurs, which cause more of A to occur, and so on. These both feedback only refer to the mechanism which inhibition or propagation occurs. Enzyme inhibitors. Inhibitors are compounds which can reduce the activity of enzyme. Enzyme may be inhibited reversible or irreversible. Reversible inhibitors may bind at the active site at some other site. Reversible inhibitors typically change VM, KM, or both. Types of inhibitors. There are three main types of inhibitors: reversible, irreversible, allosteric. Reversible inhibitors can be classified as competitive, non competitive, uncompetitive, while irreversible inhibitors classified as suicide inhibitors. Competitive inhibitors. A competitive inhibitor competes with the substrate for active site of the enzyme. A competitive inhibitor is any compound which closely resembles the chemical structure and molecular geometry of the substrate. The inhibitor may interact with enzyme on active site but no reaction takes place. They increase the concentration of substrate while decrease the chance of inhibitor binding to the enzyme. Non-competitive inhibitor. A non-competitive inhibitor is a substance that interacts with the enzyme but usually not at the active site. The non-competitive inhibitor reacts from very close to the active site. The net effect of non-competitive inhibitor is to change the shape of enzyme and thus the active site so that the substrate can no longer interact with enzyme to give reaction. Non-competitive inhibitors are usually reversible but are not influenced by the concentration of substrate, as is the case for reversible competitive inhibitors. Uncompetitive inhibition. This is a very rare case of inhibition. An uncompetitive inhibitor binds to the enzyme and enhances the binding of substrate, but the resultant enzyme inhibitor substrate complex undergoes reaction to form the product slowly.